So have you ever wondered where the cancer research that all the money that we raise is done? You know where it's done? You know where it's done here in Orange County? Let me tell you something. Right now, $8 million, more than $8 million of research funding is being utilized and put to good work at UCI. Right here. And by the way, that's about 4 times, just saying, 4 times what we raise in the county per year. So the American Cancer Society is really investing in our communities, putting that money back in here, so that great work, great cancer research can be done right here in our backyard. I have to say that a lot of the researchers that are doing that work right now are among you tonight. So I urge you to introduce yourself, find out what they're doing, and thank them for the great work that they're doing with American Cancer Society funding. So you might be asking why UCI gets so much of our money. Why do we, do we like them, or do they have an in? They don't actually, they compete for it. It's a very, very competitive situation where they have to reapply regularly for this funding. And they win it, and they win it, and they win it, and they win it. And one of the reasons they win it is a man named Edward Nelson, Dr. Edward Nelson. I have the distinct honor of introducing the man who is in part responsible for that incredible unbroken winning streak at UCI. He is our 2019 medical honoree. So let me tell you a little bit about Dr. Nelson. He's a professor of medicine and molecular biology, I have to read this, and biochemistry at UCI, and for nearly a decade has served as the chief of the Division of Hematology and Oncology. But that's not all, hold on. Since 2017, he's also been the Associate Director of Cancer Education and Training, a vital part of what UCI does in the fight against cancer, for the Chow Family Comprehensive Cancer Center. He came to UCI in 2000 from none less than the National Cancer Institute, so he knows a little bit about how the whole funding for cancer research game works. Those are titles, of course, just titles, and only a few of the very impressive appointments, accomplishments, publications, and honors that Dr. Nelson has amassed during a long career devoted to helping people with cancer live longer and better lives. Because my time is limited, fortunately for you, and Dr. Nelson would hate it, I cannot begin to list them all. Let me summarize, however, by saying simply that Dr. Nelson is a brilliant scientist, an innovative clinician, a dedicated teacher, and an enthusiastic mentor, while continuing to make his own substantial contributions to our understanding of cancer biology. He has also advanced the care of people living with cancer, most recently by initiating a first-of-its-kind supportive oncology and cancer survivorship clinic in Orange County. He has also helped cultivate the next generation of truly outstanding cancer researchers, particularly as chair of the ACS Seed Grant Steering Committee, on which I have had the privilege of serving. I can tell you that no one takes the stewardship of ACS funds and the training of promising researchers that seek them more seriously than Dr. Nelson and his steering committee colleagues. In the past 3 years, under his leadership, ACS has provided 12 pilot grants of $45,000 apiece to junior faculty. Many of these awardees go on to win the next level of extramural cancer research funding from the NIH, the Department of Defense, the National Science Foundation, and many other organizations. Dr. Nelson will shortly tell you how this amazing process works at UCI and why it is probably more effective than any other institution. In this light, you can see why Dr. Nelson is respected and beloved at UCI and at the American Cancer Society, not just for what he has done as a researcher himself, but for what he is allowing other researchers to do. It is now my distinct pleasure and honor to ask Dr. Nelson to come to the stage and accept our recognition and sincere thanks as our 2019 medical honoree.
Well, thank you very much, Tad, for the kind and generous introduction. Tonight, I don't want to talk or speak about my research or my accomplishments. Instead, I want to highlight for you why the work of researchers is so critically important to advancing the care of our cancer patients. The daunting task that they have in front of them, along with the dramatically increased pace of clinical advances. Finally, I want to share with you some of the work that comes from the ACS support through our institutional research grant and some of the extremely dedicated and gifted junior faculty scientists who have benefited and are benefiting from that support. We have a little technical glitch, so we're going to advance slides with a nod. So historically, cancer or tumors were considered to be something that were foreign to the patient or to animals. For decades, this drove the search for a magic bullet, something that would cure all of cancer. This was the basis for President Nixon's war on cancer and the search for antibodies that would identify all cancers. Those searches were by and large not very effective. In the early 1990s, with the characterization of genetic changes in cancer and the extensive studies of cancer biology, it became clear that tumors were better conceptualized as altered self. They were slight changes from normal biology. This became the foundation for the development of molecularly targeted therapeutics and an increasing understanding of the complexity of cancer. So, what does it take to be a cancer cell? The exquisite work of Dr. Weinberg and others demonstrated that there are three fundamental processes that must be disrupted to turn a normal cell into a cancer cell. The first of these is to convince the cancer cell that it's receiving a signal to grow. Now that can happen either by pushing on the accelerator or taking off the brake. Within a human cell, it's estimated that there are somewhere between greater than 200 or greater than 1,000 receptors on the cell surface that can transmit one of these signals, either the brake or the accelerator. For each of these receptors, there are dozens of molecules involved in sending the signal to the right places in the cell. You can think of them as cell phone towers and satellites that allow you to call your friends and relatives hundreds or thousands of miles away. This is an example of the number of molecules involved in the signaling pathway of the epidermal growth factor receptor family. It has 4 receptors. Don't turn your napkins over, there's no quiz. The point being that there are lots of towers and satellites to send the signal throughout the cell. So, conservatively speaking, each receptor, and let's say one of those various proteins in those signaling pathways, could be involved transmitting the signal or disrupting the signal, which would result in, say, 400 events that would result in this disruption, telling the cell that it needs to grow. The second process that must be disrupted is that of DNA repair. Now, there are 5 major repair pathways with somewhere between 20 and 40 individual proteins on each. Conservatively, we can think about those 5 pathways and maybe 6 of those molecules or proteins in each pathway as being potentially disrupted. The third process that must be disrupted is the cellular suicide. We must tell a cell that it needs to stop itself from committing cellular suicide. We call that apoptosis. There are two major pathways for apoptosis, and each of them has somewhere around 20 odd proteins that are involved in executing that pathway. So we'll again take a conservative estimate. We'll say there are two pathways and we'll take half of those proteins, 10 each. So what does that do? I mean, you have three pathways, you have a finite number of proteins. How difficult can this be? So let's look a little bit. We've got growth signaling. We got 400 events, we have DNA repair, we have 30 events. Apoptosis, we have 20 events. Now, they can be combined combinatorially. Now, for those mathematicians, here's the equation. Right? There will not be a quiz. But if you disrupt three pathways with 450 potential elements, and the next one. There are over 90 million different ways that a cell can become a cancer cell. Even with our conservative estimations, you can see that there are a multitude of ways that a cancer cell can become a cancer cell, and that is why each individual cancer is unique to that individual. If we took more or less conservative measures, we would rapidly find our number to be approaching the level of our national debt. So despite this complexity, we've made amazing progress, and I'd like to share some of that with you now. So this is a timeline of cancer therapeutics. Prior to 1940, there was essentially only one modality, that was surgery. That wasn't very successful, but of course, with the advent of aseptic technique and anesthesia, two critical elements for successful surgery, there actually were successes in the management of cancer. And then we had the development of cancer therapeutics, chemotherapy. First, the nitrogen mustards and methotrexate, and those were shown to be able to put leukemia into remission in children. The development of radiation therapy, and then combinations of chemotherapies, all happening in the 1945 to 1965 era. Then there's sort of a nadir, right? Not a lot happens between 1965 and 1985. And I want you to remember that nadir. After 1985, we start to see increasing new therapeutics, and indeed, there are 13 FDA-approved molecularly targeted therapeutics prior to 2008. So what about the 2008 to 2019? As you can see, in the first half up to 2014, that sort of trajectory upward continued. And there were more and more molecularly targeted therapeutics. That trend dramatically increased from 2014 to 2019. And indeed, there's 16 brand new molecularly targeted therapeutics in this 11-year period. That is more than 90% of the molecularly targeted therapeutics. It's 9 times the number of molecularly targeted therapeutics that were developed in the past 70 years. So this timeline is important, but what another timeline is actually understanding how those therapeutics came about. So how many of you have seen the commercial on television about Opdivo and doing wonderful things for lung cancer? Right? You know, you get to survive longer and better, all good things. In 1993, the publication of the molecule that enabled the development of the therapeutic Opdivo occurred. It was not until 2013 that Opdivo was FDA approved. That was 20 years later. Now, if you think back to that area that I asked you to remember, 1965 to 85, track back 20 years. End of World War II, Cold War, Korean conflict. Our society was not focused on cancer research. It wasn't focused on understanding the biology of normal cells and cancer cells. And as a result, there was sort of this neighbor. And once we came around and had our war on cancer declared and greater investment in the NIH, we started to see a pickup of those therapeutics, but with a 20-year delay. So with this timeline in mind, the work being started today in laboratories around the world will yield new therapies and new strategies, but for our children and our grandchildren. So I'd like to turn now to our ACS institutional research grant, which Tad spoke about. This grant has been active since 2005. It provides 1 year of pilot funding to junior faculty. Those are faculty members that are in their first 6 years of a faculty appointment. The UCI ACS IRG Award contributes 2/3 of the funds for a maximum of 4 awards per year, as Tad had mentioned, and we must renew it every 3 years. Through the generosity of the Chancellor's Office and the Child Family Comprehensive Cancer Center with the director of Dr. Richard Van Etten, who's also here tonight, they provide us matching funds that bring us up to that $45,000 number that Tad mentioned earlier. Our current 3-year award is for $360,000. That's $120,000 per year, thus the $30,000 for 4 awards. We request applications twice a year, and the applications are evaluated and competitively rated for possible funding by the ACS IRG Steering Committee. For those that aren't funded, we have instituted an intensive mentoring chalk talk. This is to improve their grant application and research project formulation skills, particularly for applicants in disciplines other than biology. I'm very proud that the steering committee developed, approved, and initiated the Chalk Talk initiative. It's worthwhile to note that we have a success rate of over 80% for individuals participating in the program and then responding to a subsequent call for applications, and a 50% success rate for those being awarded an ACS IRG award subsequent to their participation in the mentoring. So I want to highlight for you some of our ACS IRG pilot awardees. They're all in the audience, and I will ask them to stand when they're mentioned. The first is Dr. Benavente. So Dr. Benavente studies pathways that involve the protein RB. This is a very important tumor suppressor protein, and when it is mutated or lost, it results in tumor formation. Now, exactly how this loss of RB function causes all of the effects that we see on a cell is not at all well understood. Claudia is working to identify the downstream genes and pathways that are regulated by RB and have the potential then to become alternative therapeutic targets for the treatment of cancer. Although her work started in rare childhood tumors, The findings that she has discovered now look to be important in adult tumors. Dr. Benavente applied for her ACS IRG in October of 2017. It was funded. It's titled. In 2019, just a few months ago, she was awarded an ACS Research Scholar Award, and she scored very well on an NIH R01 application that we expect to be funded shortly.
Nice.
Dr. Benavente.
That's a big deal.
Her pilot award turned into $2.8 million. Dr. Daniel Chow applies his expertise. Yeah, you have to stand up.
Dr. Chow. Yeah.
Did I really skip one? I did.
I'm sorry.
All right, Michelle.
How dare you?
You have to stand up. Now, Michelle Dickman is in the Department of Biomedical Engineering in the School of Engineering. It would be reasonable to ask, what in the world is an engineer doing applying for a cancer research grant? One of the most critical characteristics of a successful scientist is persistence, confidence in themselves, their work, and their ideas. Dr. Dickman epitomizes this dedication to becoming a successful cancer investigator, as do the next two individuals that I will highlight. Dr. Dickman applies her expertise in chemistry, biochemistry, and engineering to design advanced imaging techniques to study in real time the energy dynamics of a cell as it goes through the process of invasion. She's studying how the energy powerhouse of a cell, the mitochondria, relocate and turn on when the cell starts to move. These tools and these techniques will provide invaluable insight into the process of cancer metastasis. So, I said Dr. Dickman was an epitome of persistence. This is why. She applied first in 2015, reapplied in 2016, reapplied in 2017. We invited her for the intensive mentoring, and in October of 2017, she was funded for the mitochondrial recruitment and metabolism in cancer cells, and has recently been awarded an NSF Career Development Award for that, to continue that same work. All right, now Dr. Cho, it's now your time to stand. Dr. Chow applies his expertise in artificial intelligence, machine learning, and neural networks to assess subtle changes in imaging, specifically MRI, to characterize the molecular and genetic changes in brain tumors. The genetic changes in brain tumors, or glioblastoma multiforme, or GBM as it's shortened, are important for both prognosis and determining the most appropriate treatment. His ability to identify these changes earlier than the surgical specimen and then subsequent analysis will allow physicians to know, even before surgery, what therapy should be used, expediting the initiation of therapy. Clinical research such as this inherently takes longer to come to fruition. Importantly, Dr. Chow is a practicing neuroradiologist involved in the day-to-day care of patients in addition to his research activities. Dr. Chow applied in March of 2017. He was not funded. He reapplied in October of 2017, was invited to the mentoring Choctaw, and reapplied in October 18 and was funded for the deep learning classification of glioblastoma genetic heterogeneity and survivorship. He's in the midst of his award. He has 1 year, so we don't expect him to have applied and gotten another award already. All right. Next up is Dr. Ramatapana. Farah, did
There's a reason why I refer to Dr. Patana as Farah. She studies small and large genomic data sets with a focus on genes that may help explain health disparities. In her earlier work, when she arrived at UCI, the team that she was part of found that the gene signature for prostate cancer stroma, the scaffold that holds the tumor cells, provides a very powerful signature for which tumors were likely to be aggressive and which needed earlier and more extensive treatment. Subsequently, she is focused on the role of epigenetic regulation, that is, changes in the chemistry of the DNA molecules, as a basis for this signature, and surprisingly has identified that some of the target genes were involved or are involved in the immune system. Farah applied in 2015, again in 2016, retooled, applied in 2016 and 2017, was invited for the mentoring program, and then reapplied in October of 2017 and was funded for her studies in demethylation of DNA and activation of immune system genes in the stroma as a therapeutic for African American prostate cancer, and went on to be recently awarded a prestigious ESI Merit Award. This is a 7-year award.
Wow.
Looking at the health disparities in prostate cancer between African American and non-Hispanic whites. Her award of $45,000 led her to this award of $1.8 million. Of course, all of this requires a team, and I would be remiss if I did not acknowledge the current and recently departed members of the UCI ACS IRG steering committee.
Oh!
Angela, I'm sorry.
Oh,
for two.
what? He missed another one. I was going to say, Angela didn't go away.
Dr. Fleischman is a physician scientist who focuses on She also happens to be in our division. Who focuses on care and research in a group of disorders called myeloproliferative neoplasms. These are generally slow progressing cancers, but they can evolve into acute leukemia. Her research focus has been on how the malignant stem cells, clones of those malignant stem cells, outcompete the normal blood stem cells within the bone marrow. Her work in mouse models of these diseases has identified a central role for inflammation and inflammatory signals. We call them cytokines. This has led to clinical studies of the microbiome and of the Mediterranean diet as possible modulators of these inflammatory signals. Dr. Fleischman applied in October of 2013, soon after she arrived to UCI, was awarded her IRG seed grant, has subsequently received a Vice Foundation Scholar Award, MPN Challenge Award, Department of Defense Career Development Award, and recently an NIH R21. We have asked her to serve on the ACS IRG Steering Committee, and she accepted in 2018. Her seed award generated $1.3 million. Now I get to talk about my team. As I said, all of this requires a team. And this is a group of faculty that are exceptionally dedicated to fostering the career of our junior faculty cancer researchers. They volunteer their time and effort to keep the chains moving for our junior faculty and to foster the breadth of research that can only occur at a university. That's in contrast to other cancer institutions, such as the one that has its home in Duarte. No matter how dedicated a group of faculty and ACS volunteers are, they need more. They need glue to hold them together.
Our glue starts with the Cancer Research Institute, that is the academic home of the ACS IRG, and its outstanding director, Marian Waterman. We must also acknowledge Nita Driscoll. She has a unique title. It's called Jack of All Trades. And Christina. Together, they really do help us to stay all together and keep things moving. So I would like Christina, Nita, Marian, those members of the steering committee and the awardees to please stand, and the audience please to join me in acknowledging the exceptional cancer researchers, the steering committee of ACS and the board for their tireless commitment. I hope that I've convinced you of several things. The first is that cancer is an amazingly complex disease. And thanks to research performed 20, 30 years ago, we are seeing tremendous progress in our fight against cancer today. We have phenomenally bright, driven young scientists at UCI and elsewhere, and they will continue to drive this forward with the discoveries that they are making today. Doing cancer research is not easy. It is not enough to be very intelligent. You have to be persistent, confident in your ideas, and yes, lucky enough to have good mentors. Your support of the ACS benefits patients today and patients tomorrow, the latter through the programs such as the ACS IRG. Your dollars provided to the ACS have a huge return on investment, of which you should be immensely proud, as are we for the continued partnership between UCI and the American Cancer Society. I am convinced that for nearly all individual accolades, they do not happen unless the individual so honored either assembles, leads, or is part of a very good team. I am especially proud of the teams that I am part of. Whether it's for patient care, cancer research, or cancer research education, I have great teams that I'm part of. Thus, on behalf of all the members of those teams, I want to thank you, each and every one of you, for this tremendous honor and for your ongoing, unwavering, vigorous support of the American Cancer Society. I'm deeply touched.